Thanks so much for the nice introduction, Jan. Before I begin my talk, I don't have any conflicts of interest to disclose. As Jan mentioned, I'll be discussing dietary tryptophan and its interactions with the gut microbiome and the implications of those interactions with intestinal health and systemic health as well. And I'm excited to give this talk. I think this is a really important emerging area of gastrointestinal research that has potentially major implications for both gastrointestinal and systemic health and could lead to some novel insights about the pathophysiology of gastrointestinal and systemic disorders, as well as to novel approaches to diagnosis and therapies for those conditions. So tryptophan is an essential amino acid in dogs and cats, as well as humans. Most tryptophan that we ingest or that our patients ingest, about 90% or more, is absorbed in the small intestine, where it can be either catabolized through a few different catabolic processes or used for protein synthesis. It's understood that a small portion of it, 5 to 10% or less, can escape absorption in the small intestine and enter the distal gut, the colon, where it can be further metabolized by intestinal microbiota. And those interactions between tryptophan and the intestinal microbiota impact host animal health and disease. Altered tryptophan metabolism has been identified in dogs and cats with chronic enteropathies and chronic kidney diseases. Enteric microbiota are critical to maintaining intestinal mucosal homeostasis, and emerging evidence suggests that tryptophan metabolites regulate gut and systemic health. And most of us are familiar with tryptophan when we sit down to eat Thanksgiving, or those in the US to eat Thanksgiving meal, but I hope to show that it has other potential implications for health and disease aside from thinking about it during Thanksgiving. So dietary tryptophan, there are three main metabolic pathways. For the portion that's not used for protein synthesis, the main catabolic pathway is the kynurenine pathway. And this is greater than 90% of total tryptophan catabolism in the body. There's a basal rate of kynurenine pathway metabolism in the liver, but extrahepatic kynurenine metabolism occurs in the intestines, in immune cells, and in the brain in particular. And those pathways are regulated by inflammation, interestingly. And it has the potential to generate neuroactive compounds that participate in regulation of the gut-brain axis. Another important pathway is the serotonin pathway. Through this pathway, tryptophan is catabolized into 5-HT, which is serotonin. This primarily happens in enteroendocrine and enterochromaffin cells within the intestinal tract. And interestingly, about 90% of all bodily serotonin is derived from the gut. And the synthesis of serotonin from tryptophan is highly influenced by enteric microbiota in conditions in the gut lumen. And multiple studies have shown that serotonin interacts with the enteric nervous system and the autonomic nervous system to regulate both intestinal health and gut-brain axis interactions. And then the final pathway is the microbial indolic pathway. This pathway is responsible for about 5% of dietary tryptophan catabolism. This represents the fraction that escapes absorption in the upper gut and makes it into the colon, where it can be metabolized by bacteria. And it has really diverse effects on host health. Studies have shown that these compounds have immunomodulatory properties. They support mucosal barrier function. They regulate metabolic health. They have a variety of really important effects in the host animal. And collectively, these are called microbial indole catabolites of tryptophan. So taking a closer look at the kynurenine pathway, this is a very fascinating pathway that's involved in the gut-brain axis regulation. Tryptophan that is absorbed through the intestinal epithelium can be metabolized or catabolized further into kynurenine. Kynurenine is able to freely cross the blood-brain barrier, where it can enter a few different catabolic cascades, some of which are regulated by inflammatory processes. So we see the red star. These are pathways that are upregulated during states of inflammation. So kynurenine can flow down and result in the generation of quinolinic acid and anthrolinic acid. And these are really important metabolites that have been identified as having neurotoxic properties. For instance, quinolinic acid is an NMDA receptor agonist. And Alternatively, when there's not a pro-inflammatory state, you can have the generation of kynurenic acid, which has neuroprotective properties. So here we see the balance of tryptophan catabolism has potentially significance in regulating gut-brain axis and regulating behavioral states as well as brain health. And this is relevant to how the gut and the brain interact to regulate neurologic health. The next pathway, again, is the serotonin pathway. So again, this is mainly occurring in enteroendocrine and enterochromaffin cells within the intestinal epithelium. This process is stimulated by short-chain fatty acids, which of course are products of microbial metabolism. The short-chain fatty acids stimulate these enterochromaffin cells to convert tryptophan into serotonin. And that serotonin can be released basally into the submucosa, but also apically. And interestingly, the apically secreted tryptophan has a role in promoting colonization by commensal microbes that are important to support host health. For instance, Therapeutic, which is a short-chain fatty acid metabolizing species, which has been found to promote intestinal health, is supported by the presence of its growth is supported by the presence of tryptophan in the gut lumen. Also, tryptophan is released into vagal afferent nerves, which have anti-anxiety and anti-depression effects and can affect, again, this concept of the gut-brain axis. This is one way in which the gut has a direct line of communication to the brain. Tryptophan that enters circulation also has effects on metabolism. It increases insulin secretion and promotes beta cell survival in the pancreatic islets, and can support hepatocyte regeneration, and has interesting kind of effects on stellate cell activation and fibrosis, as well as gluconeogenesis. And then locally, tryptophan interacts with the enteric nervous system to support intestinal motility, and it generally has the effect of increasing intestinal motility. The third pathway, and the one that I personally find to be the most interesting, is the microbial indolic pathway. So collectively, these chemicals that are produced by gut microbes from tryptophan are called microbial indole catabolites of tryptophan, also called MICs. It's important to recognize that these are, and one of the poster child for this family is indole-propionate or indole-propionic acid, which I've shown in the chemical form up in the upper right. And it's important to understand that these are exclusive products of gut microbial catabolism. There was a study where germ-free and conventional mice were compared to one another. So conventional mice have a normal mouse microbiome, and germ-free mice were basically mice that did not have a microbiome. They were grown in pathogen and germ-free conditions. And what we see is a total lack or absence of indole-propionic acid in the germ-free mice. And that suggests that they have bacteria as their exclusive source. Numerous bacterial species can catabolize tryptophan into these MICs, and multiple studies have shown that MICs regulate intestinal homeostasis, immune responses, organ system health, and energy metabolism. So this is a really simplified kind of schematic of the tryptophan degradation pathways by bacteria. Commensal organisms essentially control the flow of tryptophan through these different pathways. Bacteria that are associated with a state of eubiosis in health promote, and we're talking about lactobacillus and bifidobacterium primarily here, they can promote the conversion of tryptophan into these MICs, indole-propionate, indolactate, and indolaldehyde. And these are known to have beneficial effects on gut and systemic health. Bacteria associated with dysbiosis, including E. coli and some Clostridial species, convert tryptophan into indole. And indole is further metabolized in the liver into indoxyl sulfate, where it can potentially have nephrotoxic properties and promote the progression of renal injury and chronic kidney diseases. And this is an interesting perspective, because here we have molecules that on one hand have beneficial effects in the host, but on the other hand potentially have deleterious effects to systemic health. And we have enteric microbes situated at the nexus of controlling which way the tryptophan will flow. Will it generate beneficial compounds, or will it generate compounds that are potentially deleterious to host health? This is just showing a variety of potential systemic and local effects of these MICs using IPA as the poster child. They are shown to have antifibrotic effects in the liver. They promote intestinal homeostasis, which I'll talk more specifically about in a moment. They have anti-inflammatory properties. Indole-propionic acid in particular is an extremely powerful antioxidant molecule with neuroprotective properties. They're shown to be anti-tumorigenic in animal models and regulate metabolism and generally have an anti-obesity effect as well. So you can see not only do these compounds generated by bacteria have local effects on intestinal homeostasis, but they affect systemic health through a variety of different mechanisms. A really interesting study, a couple studies have come out recently, including one from a collaborator and friend of mine, Stacy Summers, that going back to this concept of indoxyl sulfate, it's been shown that indoxyl sulfate is increased in the serum of dogs and cats with chronic kidney disease and actually increases in concentration as they progress through the stages of chronic kidney disease. And Stacy Summers, who's now at Oregon State, has also shown that the increase in the indoxyl sulfate is associated with decreased microbiota diversity in cats with chronic kidney disease. So again, even though these molecules have a lot of potential benefits in the host, understanding more about how to direct their metabolism toward beneficial compounds versus things like indoxyl sulfate will be important for understanding their role in health and disease and whatever therapeutic potential that they may have. So focusing more in on the local effects of these indolic compounds, they've been shown to regulate intestinal homeostasis by promoting immunomodulatory responses and strengthening the mucosal barrier. So here in a state of eubiosis on the left, where we have a rich, diverse community of commensal microbes that are capable of converting tryptophan into these indolic derivatives, these MICs, these compounds are freely absorbed through the intestinal mucosa, where they can interact with the host tissues and in the state of health through the pregnane X and aryl hydrocarbon receptors. They're known to increase the expression of tight junction proteins, increase mucin, so a thicker mucus layer, and increase the secretion of antimicrobial peptides, which have a regulatory effect in controlling luminal microbial populations. And then signaling through leukocytes, immune cells in the submucosa, they're also found to increase the expression of IL-10, which is an anti-inflammatory immunomodulatory cytokine that suppresses pro-inflammatory responses. When we contrast this with the state of dysbiosis associated with many chronic enteropathies, we have a loss of some of these commensal organisms that catabolize tryptophan into these MICs with lower microbial diversity, and we lose a lot of these important interactions. You have decreased expression of tight junction proteins, mucins, and antimicrobial peptides. You have secretion of pro-inflammatory cytokines like TNF-alpha, and then you have lower expression of IL-10, leading to dysregulation of inflammatory responses and gut mucosal inflammation. Recently, along with some collaborators, I performed an investigation looking at serum concentrations of these MICs in cats with chronic inflammatory enteropathy and low-grade intestinal T-cell lymphoma and compared them to healthy controls. And as you can see, concentrations of tryptophan were significantly lower in the serum of cats with chronic inflammatory enteropathy and low-grade T-cell lymphoma, as well as indole propionic acid, indole acrylate, indolactate, indole pyruvate, and indolaldehyde. So these are statistically significant effects showing in this population of cats with chronic enteropathies lower concentrations of these indolic derivatives. Further, we found that these serum MIC concentrations were inversely correlated with the feline chronic enteropathy activity indices, suggesting that alterations in tryptophan metabolism are associated with more severe disease states in these cats. And this is really similar to findings in humans with inflammatory bowel disease as well. We also looked at full-length 16S sequencing to characterize the microbiomes of these cats. And you can see here, and just for reference, this is 0 here. So everything above 0 is increased, everything below 0 is decreased. And it's a log 2 scale, meaning that this is 2 to the negative 25th power of a difference. So these are big differences, not small differences. So we saw multiple species of bifidobacterium that were significantly decreased in the feces of cats with chronic inflammatory enteropathy and low-grade intestinal T-cell lymphoma. We also found that the abundance of these organisms was positively correlated with the concentrations of MICs in the feces, suggesting that, as it has been characterized in other species, that bifidobacterial species it participate in the catabolism of tryptophan in these cats. We also wanted to investigate the mechanistic impact of these indolic derivatives using a canine intestinal organoid model. So intestinal biopsies, colonic biopsies were taken from healthy dogs, and they were grown in a monolayer, a polarized monolayer, which simulates the gut environment. The apical chamber is the luminal side, the basal chamber is the submucosal side. We treated them, we pre-incubated them with indole acid, and then later expose them to an inflammatory stimuli using TNF alpha. And we use transepithelial electrical resistance to measure the basically as a proxy for intestinal permeability. So the higher the tier value, the lower the intestinal permeability. And then we characterize gene expression analysis to understand what type of pro-inflammatory or anti-inflammatory responses were present in the cells that were treated with IPA. So we found some really interesting effects, and there's so much data from this experiment, so I'm just showing a very tiny snapshot. First, we saw that the collonoids that were treated with TNF-alpha had a significant reduction in the tier value, suggestive of increased intestinal permeability. Pre-incubation with indole acid totally normalized tier in response to the TNF-alpha stimulus. So it totally blunted the effect of inflammatory stimuli on intestinal barrier function. And then looking at a really important receptor for IL-10, which is present on intestinal epithelial cells, TNF-alpha significantly reduced expression of that receptor, whereas pre-exposure to IPA before stimulation with TNF-alpha basically reversed that effect and increased the expression of the IL-10 receptor. And if you recall from a few slides ago, IL-10 is an important immunomodulatory anti-inflammatory cytokine. So we think that this could increase the responsiveness of the intestinal epithelium to anti-inflammatory signals. And this is really similar to other studies that have been done using human cell lines and in mouse models as well. We also found that genes that were upregulated by IPA regulated T cell proliferation and promoted intestinal epithelial cell development. And genes that were downregulated by IPA are those cytoskeletal type of processes involved in rearrangement of tight junctions and altered cell permeability. So we think that this showing in canine cell models really interesting mechanistic effects of these indoles that are consistent with research in humans and rodents. So there's a great promise in the idea of modulating these tryptophan catabolic pathways for therapeutic benefit. However, this pathway hasn't really been well studied in dogs or cats. There have been studies of the effect of tryptophan supplementation on behavioral disorders in dogs, but those have had mixed results. But there are a variety of preclinical studies showing a very interesting potential benefit of modulating tryptophan catabolic pathways for therapeutic effects. And there are a few different strategies that could be employed here. One would be simply dietary tryptophan supplementation. Numerous studies in mouse models have shown that excess supplementation above dietary requirements of tryptophan increases microbial production of these microbial indolic derivatives and can actually ameliorate mouse models of colitis, just tryptophan supplementation by itself. Fiber and prebiotics also have the potential promise of altering tryptophan catabolism, probably through complex interactions among enteric microbiota and shifts in microbial metabolism. Probiotics, so giving probiotic bacterial species that are known or are engineered to participate in tryptophan catabolism is a promising approach. And then the concept of postbiotics, so just taking the actual product of microbial metabolism, the mix themselves, and providing them as a therapeutic. And then there's some really interesting novel drug candidates that interfere or alter the tryptophan catabolic pathways, and I'll review one of those briefly in a moment. So starting with dietary tryptophan supplementation, there have been some studies in dogs showing interesting effects of dietary tryptophan supplementation. One showing the effects of, this was from Kelly Swanson, I know Sarah presented, who's in his lab, presented some other research coming out of that group, but showing the effects of dietary tryptophan supplementation on stool quality, fecal metabolites, and body condition composition and training sled dogs. Sled dogs are an interesting model because a lot of them have increased or poor stool quality in association with their training. I think the athletic demands put a lot of stress on their intestinal tract. And we can see here, compared to the control group that was fed a placebo, the treatment group had a lower proportion of animals with higher fecal scores. And as the fecal score increases, the stool quality decreases. So a fecal score of 5 is diarrhea. A fecal score of 1 or 2 would be considered more normal. So we see a lower proportion of animals or dogs in this study with high fecal scores in the treatment group compared to the control group, suggesting that they are capable of improving stool quality, and that was a statistically significant effect. Tryptophan supplementation has also been found in dogs to increase the circulating concentrations of circulating concentrations of serotonin, showing again that interaction with the serotonin pathway in dietary supplementation with tryptophan. Several studies have been done evaluating the effect of tryptophan on canine behavior. As I mentioned earlier in the slide deck, tryptophan has interesting potential interactions with the gut-brain axis that can influence behavior. And the results have been mixed. I just pulled a sample of a few of these studies to show here. In dogs with privately owned dogs with mild anxiety, and then in response to stimuli from familiar or unfamiliar individuals, there's been no significant impact of tryptophan supplementation on behavior. And then other studies looking at dominance and aggression behavior and anxiety have shown reduced aggression and anxiety in pet dogs or sled dogs that were fed tryptophan as a dietary supplement. There are other studies using supplements that contain other ingredients in addition to tryptophan, which have shown either a benefit or no benefit, but I don't review those because it's difficult to tease apart the effects of those other ingredients versus just the tryptophan in isolation. So another thing to consider that I mentioned earlier, if we're going to explore the potential therapeutic benefit of tryptophan, is how we can shift tryptophan catabolism away from indole, because indole is a precursor for indoxyl sulfate, which is potentially nephrotoxic, and shift it toward these other indoles, which have beneficial effects and are not involved in further catabolism to nephrotoxic or compounds that have potential deleterious effects. So this study found that tryptophan, along with pectin, a fiber source, is able to shift the balance of tryptophan metabolism by decreasing the production of indole and promoting the production of these other MICs with more beneficial properties. So here, these are just looking at 2 different concentrations or doses of tryptophan with minus or plus, or with plus or without minus pectin. And we can see the indole concentrations in these patients were significantly decreased by supplementation with pectin, regardless of the tryptophan dose. And then when we look at the indolactate, which is one of the beneficial compounds, we see that in the high dose tryptophan group in particular, we saw increased production of indolactate. And this was done in an in vitro fermentation model. But again, it's evidence that you can shift through simple dietary intervention, shift the balance of tryptophan catabolism and influence it to produce compounds that have beneficial effects or potential beneficial effects in the host. There are also some studies in mouse models looking at probiotics. MICs produced by L. reuteri, which is a really interesting potential probiotic species of lactobacillus, have been found to protect against inflammation and mitigate enteric microbiota dysbiosis in a mouse model of colitis. So this is a DSS mouse model of colitis. So the DSS group were mice in which colitis was induced, and then the DSS 15007 group was the group that were administered the probiotic lactobacillus. And this is indole carboxyaldehyde, indole acetate, and indole propionic acid here on the y-axis. And we can see, compared to the controls, the colitic mice had decreased production of these all of the indoles, and then administration of the probiotic basically rescued that to something similar to the normal state for indole carboxyaldehyde, indole acetate, and significantly increased the production of indole propionic acid. So this is an example of how, by giving a potential tryptophan catabolizing species as a probiotic, we can shift tryptophan catabolism to the production of these potentially beneficial compounds. And this was also found, if you look at B and C on the right, the L-reuteri administration was also found to decrease expression of inflammatory cytokines, including IL-6 and IL-1 beta in the colonic mucosa of DSS colitis mice. Finally, the concept of using tryptophan derivatives as a postbiotic and just synthesizing them as chemicals and giving them as a postbiotic compound is also an interesting approach. And there is a study from 2018 showing that oral endopropionic acid administration ameliorates DSS colitis in mice and reduced cytokines, inflammatory cytokines in this mouse model. So here we have the mice that were treated with DSS compared to those that were not treated with DSS. You can see there's an abundance of inflammatory infiltrates in the mucosa. And the mice that were fed endopropionic acid, you can see there's significant reduction in the inflammatory infiltrates in the submucosa. And this is also shown in the significant reduction in the histologic score comparing the DSS colitis mice to those that were also administered endopropionic acid. And what's really fascinating about this to me is that IPA, endopropionic acid, recently underwent phase 1 safety studies in humans. It was a target drug candidate for treating Alzheimer's and other neurodegenerative disorders due to its due to its free oxygen radical scavenging properties, its antioxidant properties, but also inhibits the formation of beta amyloid plaques in the brain. So it was explored as a drug candidate for Alzheimer's, and in phase 1 studies, it was very well tolerated in humans. So I think there's a lot of potential in using these compounds potentially as postbiotics. And then a somewhat more recent study showing the development of a drug inhibitor that actually inhibits the tryptophanase enzyme, which converts tryptophan into indole. And again, indole is a precursor for indoxyl sulfate, which is nephrotoxic. And this molecule, 3S-ALGO5, reduces the cecal indole load by blocking this microbial enzyme from producing indole and decreases serum concentrations of indoxyl sulfate with minimal impact on other tryptophan catabolites. So again, here we have an example of a drug candidate that interferes with some of these microbial pathways of tryptophan degradation to push things in a direction to improve health. So to conclude, we have talked about how tryptophan metabolism through the kynurenine, serotonin, and microbial indolic pathways are important to maintaining gastrointestinal, systemic, and behavioral health. Altered tryptophan catabolism has been identified in dogs with chronic enteropathies and dogs and cats with chronic kidney disease. Therapeutic interventions targeting tryptophan metabolism are promising for the treatment of gastrointestinal, systemic, and behavioral disorders. We have a few ongoing research efforts in my lab. We're looking further assessing the mechanistic role of these MICs on intestinal barrier function and inflammation. And this winter, I've got a clinical investigation looking at the impact of a bifidobacterium-based probiotic on microbial tryptophan metabolism in cats with chronic enteropathy. And that will start again in this winter in January. With that, I'll conclude, and I'm happy to take any questions.